Happy New Year and welcome to the January 2023 meeting of the Linnean Society of New York. I'm your president, Rochelle Thomas, and will now call the meeting to order. I want to remind everyone to save the date, March 14th, when we will hold our annual meeting in person at the Liederkranz Club for the first time in 3 years. And as I mentioned last month, our annual meeting is open to members and their guests only. So if you've neglected to renew your membership or are interested in joining, now is the time to do so. Our board has been hard at work, asking the perennial question of, what can my Linnean membership do for me? As such, we are in the final stages of preparing a member survey that we hope to send to all of our members within the next few weeks. So members, please check your inboxes and send us your thoughts so we can make sure the society is serving you. I now have the sad job of announcing the passing of past president, Ellen Messer. Ellen Messer served as recording secretary for the society twice, from 1999 to 2001, and then again in 2003 to 2005, before serving as the president from 2005 to 2007. His artwork graced many Linnean publications, including an issue of Proceedings. He was a field trip leader and also happened to be my own personal Linnean Society sponsor. He will be missed by many in our community. Tonight, as always, we've disabled the chat feature, so if you have questions, please type them into the Q&A box. Our Vice President, Gabriel Willow, who, as is the norm, will soon be embarking on an incredible adventure, this time to South America, but is here tonight to answer your questions at the conclusion of the lecture. On to business and our monthly voting results. Motion number 1, to accept the following members of the Linnean Society passed unanimously with 178 in favor and 0 opposed. New members and sponsors are Samari Weinberg, sponsored by Karen Asakawa, Beth LaBouche, sponsored by Ken Chea, Corey Farwell, sponsored by Ken Chea, and Ian Bell, sponsored by Miriam Murkowski. Welcome to our newly elected members. Motion number 2, to accept the December 2022 meeting minutes, passed with 176 in favor and 2 abstentions. And now I have the honor of introducing Dr. Meredith VanAcker. Dr. Meredith VanAcker is a disease ecologist currently working as a George E. Birch postdoctoral fellow under the Smithsonian's Global Health and Movement of Life programs. Her work integrates methods from wildlife movement and ecology to examine how animals' responses to land use change impact the distribution and prevalence of zoonotic pathogens and infectious disease risk for humans. Dr. VanAcker completed her PhD at Columbia University, where she worked with wildlife populations in New York City to determine the ecological drivers of urban tick-borne disease risk. She now examines multi-species movement and contact to determine how these interactions determine risk for pathogen spillover in Nairobi, Kenya. Dr. VanAcker also happens to be someone who was once my own TA for a mammalogy class at Columbia. Tonight, Dr. VanAcker will present wildlife movement and infectious disease emergence in complex ecosystems. So over to you, Dr. VanAcker. You're muted, just so you know.
Thank you so much for the introduction, Rochelle, and I'm so excited to be here tonight. I'm just going to share my screen, so bear with me for just a minute. Okay. Hi, everyone. I'm very excited to be here tonight. So I am Meredith. I'm a disease ecologist, and I'm a postdoc fellow with Smithsonian's Global Health Program. As Rochelle mentioned, I did my PhD at Columbia, so I'm really excited to share some of the local insights on the effects of wildlife movement on emerging infectious diseases. So first, I'd like to review some terms that we use in disease ecology, and this is because they might be referenced in the talk, although the last few pandemic years have unfortunately increased the literacy in this arena quite a bit. So the first is infectious disease. These are illnesses caused by pathogenic microorganisms, including bacteria, viruses, parasites, or fungi that can be spread directly or indirectly from one person to another. The second is zoonosis. This is an infectious disease that can be transmitted from animals into humans. And then a zoonotic spillover event. So this is describing that transmission from vertebrate animal to human. Vector is an organism that transmits a pathogen from one organism to another. And these are typically thought of as mosquitoes or flies or ticks. And then reservoir host. So this is a reservoir of an infectious agent where the agent lives, grows, and multiplies within that host. And infection is typically asymptomatic or non-lethal to the host. Okay, so now I'll move on to talk about the global burden of zoonotic diseases. So what's the current status and where are we heading? The majority of human emerging infectious diseases are zoonotic. It's estimated that 71% of the infectious diseases that we experience or their origins come from animals. And emerging zoonoses going into the future are most likely to be viruses. This is because of their high mutation rate, and that allows for cross-species transmission and adaptation. A recent study that was done in 2022, so shortly after COVID started, estimated that there's an annual mortality from viral disease epidemics of 3.3 million lives. And this is estimated with our current global population in mind. They also looked at what would happen if we put into place prevention strategies that could reduce the likelihood of outbreaks. And they found that even reducing outbreaks by 10% can reduce the expected deaths by 300,000 people, and also save up to $2 trillion USD annually. So this is just to, not that we need a reminder, but also to note that on top of these human losses, there are significant economic impacts from these zoonotic outbreaks. And then importantly, massive inequities in access to healthcare and the surveillance of infectious diseases exist between high and middle to low income countries. And this exacerbates the burden of zoonotic diseases. So they're very important to keep in mind, both in terms of our global public health, as well as the stability of so many of humanity's systems. And where are we heading? So researchers have attempted to predict future risk of zoonotic disease emergence. This heat map's displaying one example of predicted emerging infectious disease events, and they used a global database and predictors that span human activities like deforestation, agricultural intensification, as well as animal characteristics like biodiversity and phylogenetic proximity. And then they include environmental factors. So as you can see, the areas in yellow are showing the areas with the highest risk, and these overlap with forested tropical regions. And so these are areas where biodiversity is high and land use change is high. And land use change, as I'll explain in the coming slides, is a very powerful impetus behind disease emergence, although it's not the only driver. So there's various pathways to zoonotic spillover. And again, these are the pathogens that are moving from animals into humans. These pathways describe basically the ecological interactions that underlie the spillover event. So I'll start from the top of this figure and kind of we'll work our way down. So first at the top, we have the distribution of hosts, of reservoir hosts, and the intensity of infection within those hosts. So these hosts can be livestock as well as wildlife species like rodents, primates, bats, or birds. And this describes where these animals are distributed on the landscape, their densities, and their infection status. And then the pathogens that these animals may be carrying will then be released from the reservoir host. And they can be released through natural excretion, so urine, saliva, poop. During slaughter, the pathogen can be released when humans are handling the animals and slaughtering the livestock. Or the pathogen can be released through a vector-borne release, where a vector, like a mosquito or a tick, obtains infection when they feed on the host. And the pathogen release also describes the pathogen's survival and its movement. So next we have the human exposure element, and this is where human behavior plays a really large role. So humans can be exposed to pathogens in the environment through the butchering or eating of infected meat, or through a bite from an infected vector. And then lastly, the susceptibility of the human host can determine whether that pathogen successfully kind of colonizes the human, so whether the person actually becomes successfully infected, and the future spread of that pathogen. So this is all of these kind of ecological interactions, again, are happening pre-spillover and spread in the human population. So now that we know these underlying factors, I want to just emphasize the importance of timing. So all of these factors have to align both in space and time for spillover to occur. So this is wonderfully shown in this animation that scientist Rena Plowright produced with her team, where all of the factors are kind of shifting around in space, like little pieces of Swiss cheese, and then when the holes all overlap, spillover can occur. So there's a space element as well as a time element. And then we have environmental changes that impact these pathways and can accelerate spillover events. So land use change is one of the most dominant ways that the pathways can be disturbed. They can alter the distribution, land use change can alter the distribution of animals, as well as change the human exposure to the pathogen, because as you can imagine, if a forest is converting into an agricultural field, human behavior may change along with that change in land use. Another big disturbance is climate change. So climate change can alter the pathogen development, as well as the shedding rate from the hosts, and change pathogen pressure. Climate change has also been shown to alter the range and survival of mosquito vectors, which can then change mosquito-borne disease dynamics more broadly. Agricultural intensification is another major disturbance that changes pathogen infection dynamics because it inflates animal densities when you have a lot of livestock kind of put together in a smaller space. And it presents more opportunities for human exposure through slaughter. But agricultural intensification can also increase the amount that humans are encroaching into natural areas like forests. And lastly, we have wildlife trade and translocations. So wildlife trade can import both known and unknown pathogens into naive populations. And this alters the pathogen movement and distribution in space. So out of the different disturbances that I've discussed here, I'll be focusing on the impacts of land use change specifically on zoonotic spillover. So one of the most dominant types of land use change is urbanization. This urban growth dramatically transforms the landscape. So hopefully this works. We have a time series here showing The expansion of a city in China, Dalian, that is 7.5 million people. And you can see over time how even the extent of the city is expanding into the waterways. So currently, cities are home to 55% of the world population, and they continue to increase and are expected to increase to 68% of the global population. 68% of the global population will be in cities by 2050. And while high-income countries tend to have the infrastructure to support this growth, lower-income countries are facing extreme developmental challenges in managing these growing human populations. So a consequence of this is inadequate planning, poor sanitation, and poor infrastructure that together can undermine aspects of public health. And I'd like you to just kind of keep that in mind as I go through these two case studies. The first will be from here in New York, a developed city with a lot of financial support, and a rapidly developing city in Nairobi, Kenya. And overall, land use change in its many different forms creates these novel contact and encounter opportunities between species that may not have ever encountered each other in the past, or are now frequently encountering each other in new ways. And so this presents pathogen transmission opportunities that may not have existed previously. So urbanization in particular can facilitate zoonotic disease emergence and spread through land use alteration, environmental stressors, and human population dynamics. The fragmentation of habitat changes what wildlife species can persist on the habitat and then where they can move. And then environmental stressors like food scarcity or clustering of wildlife around resources because they've become more limited in an urban environment can increase their susceptibility to infection. With land cover conversion, humans may come into contact with animals more frequently along edge habitats, potentially increasing the transmission probability. And then these last 2 points we've seen firsthand with SARS-CoV-2, where high human populations in cities lead to this rapid pathogen spread through a population. We've seen this firsthand in New York, and then that the connectivity of people between cities globally contributes to these extremely fast variant introductions to new areas. So there are diverse socio-ecological drivers of these zoonotic hazards within a city that can be unique to a particular place. So these drivers include biotic interactions. So these are interactions between organisms. And this image is showing that here with the green bubbles. So we have things like resource availability and feeding, species movement, behavior and immunity, host community diversity, and then species invasion. And then there's also abiotic conditions that can drive these hazards. So climate change, urban heat island effect, the imperviousness of the surface and kind of the hydrology regimes. And lastly, the landscape characteristics can also help to determine what zoonotic hazards exist. So this includes things like patch composition and configuration, as well as socioeconomic inequalities that emerge as physical characteristics on the landscape in terms of inequities between housing structures and resource distributions. So these socio-ecological drivers are underlying the zoonotic hazards in cities. And here the zoonotic hazard is kind of the combination of the host and vector distribution and their abundance, as well as their pathogen prevalence. So if we think back to those pathways of spillover, it's kind of that first box on the top where we're thinking about where are the animals and what are they infected with. So when we think about the zoonotic hazard in combination with human exposure and human vulnerability, that determines the spillover risk in an urban area. And just to give some context, this paper was put together by my previous lab at Columbia. And so, you know, when we were kind of coming together thinking about zoonotic hazards, New York City was really the model for this paper, and as well as these kind of socio-ecological drivers. So I'd like to just continue to use this figure to narrow us in on thinking about how urban environments may impact animal movement. So I've highlighted just kind of the bubbles that continue to be, you know, really relevant for wildlife in cities. This is patch composition and configuration. So what are the habitat patches? How are they shaped? How connected are they? Are they forested? Are they a combination of shrubs and residential? So all of that kind of matters as animals are trying to move across the landscape and find cover, forage, and each other to successfully reproduce. But we also know that wildlife barriers and corridors play a large role in the movement of animals in cities. And then resource availability and feeding. So there's artificial feeding from people, and then resource availability coming both from natural sources and anthropogenic sources. So things like trash, gardens, ornamental plants, that sort of thing. So all of these in combination are going to affect the way that wildlife move through the city and the impact that they have on zoonotic diseases. And host movement can have kind of different impacts on disease systems depending on how that pathogen is transmitted from reservoir host to recipient host. So this figure is showing how pathogens can overcome species barriers in different ways to result in spillover. And there's 4 kind of different models here. The top panel shows a pathogen moving directly from the source host to the recipient host. So an example of this would be a pathogen like rabies lyssavirus, where a rabid animal directly bites a human and transmits the virus. Panel B shows a scenario where a pathogen is transmitted to the recipient host through an intermediate host that's a vertebrate. And livestock often serve as these intermediate hosts. If you're familiar with Nipah virus, this is a virus that's transmitted from bats, frequently into pigs, and then from pigs into humans. In panel C, we see a similar model, but that intermediate host is an invertebrate. So this is describing a vector-borne disease scenario where a tick or a mosquito will vector the pathogen between the reservoir source host and the recipient host. And then lastly, in panel D, we have a pathogen moving from a reservoir into the environment and then into the recipient host. So this could describe something like E. coli, which can persist within the environment. And today I'll be going over 2 case studies that describe the models from B, C, and D. And I'll start by describing the scenario for vector-borne disease in New York. So New York City has experienced increased tick-borne diseases. So this is a vector-borne disease that's also zoonotic, or this is a group of zoonotic vector-borne diseases. And this increase has happened over the last 20 years. This is shown on the graph on the right here, where there's an axis for the number of tick-borne diseases excluding Lyme, and then Lyme disease. And that's because the cases for Lyme are greater. in the city when compared to the other Lyme disease cases. The most prevalent tick-borne diseases in New York are babesiosis, anaplasmosis, and then Lyme disease. And all three of these are vectored by the black-legged tick, Ixodes scapularis. So we're left to wonder why cities have experienced this increase in tick-borne diseases, because New York is not alone in this. And one hypothesis is that the greening of cities supports the movement of wildlife into urban areas. And along with that brings their pathogens and vectors that are introduced along with the wildlife. So briefly, I'm going to just point us to one particular point of interest in terms of the life cycle of the black-legged tick and the transmission cycle of Borrelia burgdorferi. So if you bring your attention down to the bottom, this is where the adult black-legged tick takes a blood meal from deer. A large final blood meal completes the tick's life cycle because it allows the female to lay egg masses. And deer are really essential for that. They feed up to 500 ticks. One deer can feed up to 500 ticks, and then each tick can then lay up to 2,000 eggs. So this makes them really important for tick population establishment. And it's important to note that the tick only moves 1 meter vertically in space. So dispersal over longer distances takes place when it feeds on a host. And this aspect of the black-legged tick's natural history is really what explicitly links the deer movement with tick-borne disease, because deer don't transmit the bacteria, Borrelia burgdorferi, to feeding ticks, but they move and maintain tick populations. So you can kind of think of them as tick subways for all of us New Yorkers. So this map is showing citywide Lyme disease risk that we estimated in 2017 to just provide some context. The size of the circles show the density of nymphs, and the color of the circles shows the nymphal infection prevalence, where the darker circles have higher infection with the bacteria. And the highest Lyme disease risk was located on Staten Island. And so this is where the research examining the impacts of deer movement on tick distribution was focused. Okay, so how exactly can white-tailed deer move or alter tick-borne disease risk? As I mentioned, when you think back to that figure with the little blurbs, resource selection is a big driver of why animals move. So deer resource selection of anthropogenic features, things like ornamental plants or people's gardens, can bring feeding ticks in closer proximity to humans and potentially increasing their exposure risk. Another way is through seasonal movement. So seasonal movement patterns for deer can align with the seasonal activity cycle for ticks and create life stage specific dispersal events. So to explain this a little better, hopefully this plot helps. It's showing the movement trajectory of one individual from Staten Island, one individual deer. This z-axis is time. So this is over the course of 1 year. And then the X and Y is showing the latitude and longitude. So where is the animal in space? And you can see that there's a shift in the spatial location of the animal over the course of the year. So if you could imagine that a deer is in a habitat patch with adult ticks that are questing, those ticks attach onto the deer and the deer makes the shift over space. Then the ticks drop off and lay their eggs. That can help to disperse the ticks over space, as well as potentially affect the population dynamics of the ticks. And then individual deer traits may affect tick burdens and movement behaviors. So some individuals may be less risk averse, or we might see sex-based movement differences. that can make one sex more impactful. So this is showing deer home ranges from northern Staten Island of male white-tailed deer and female deer. And you can see that there's a massive size difference between the few females and between the males and females home ranges on Staten Island. And then finally, deer movement behaviors, things like speed and directionality in different habitats can affect their contact rates with ticks. So this diagram is showing a trajectory where sections of the path are more linear, and then there's other sections that show a lot more angles. And so if the animal is moving faster in a more linear way through this habitat, but taking more time and spending more time turning in this other habitat, they may have a higher chance of coming into contact with ticks when they have this high tortuosity movements. Okay, so to explore some of these relationships, I worked with GPS data. This was in collaboration with New York City Parks and White Buffalo. And following the data cleanup, I will show results from 50 males and 9 females that were included. And then we also sampled for questing ticks collected from residential areas on Staten Island between the same time periods. Okay, so first we found, similar to what that previous slide showed, that male home ranges on average were much larger than females. The average home range for males was 170 hectares, whereas the average for females was 50 hectares. And you can visually see that difference in this map on the left, where the home ranges for males are shown in blue and the home range for females are shown in white. And then we found that as home range size increased, the percent natural land cover within the home range decreased. So this is shown by this plot on the top, where home range is on the y-axis and the percent natural areas is on the x-axis here. So as natural area decreased, the habitat diversity within the home range increased. So this pattern indicates that deer with larger home ranges may not be finding adequate resources within the habitat patches that make up the core of their home range, and that they have to move beyond kind of that core in order to obtain the resources that they need. And then when we looked at deer movement and direction over urban and natural habitats seasonally, we saw that, well, first let me just explain some of these plots. We have male deer on the top row here, female deer on the bottom row. And then each of these boxes is a different breeding season, or a different season for the deer. So we have pre-breeding, breeding, and post-breeding. On the, and then the x-axis is showing natural and urban land cover. So we found that both males and females moved fastest through natural land covers, and males moved the slowest over urban land covers during that pre-breeding season, whereas females moved the slowest during the breeding season over urban land covers. So this is important for, again, thinking about the overlap with the tick seasonality. So if females move the slowest during the breeding season, this could potentially distribute feeding adult ticks, because that's when they're most active, moving from natural to urban land covers. And then the pre-breeding season for males, this is when nymphs are most active. And so this would spatially overlap with nymphs trying to find hosts. And because we were interested in how deer might use space around people's homes, so whether they're attracted to backyards specifically that could make for people encountering infected ticks more likely, I put together this layer that describes different residential blocks. And I won't go into details, but this is basically just to show that there's these two different types. One's really forested and more connected for deer to access, and one is higher impervious surface and less connected for deer. Oops, go back one. Okay, and what we found with our models is that deer increased avoidance with higher development intensity, but they made selections between these two block types where they preferred habitat within the residential blocks that were more forested and more connected. They selected that over the block types that were more impervious and less connected. And then the take home here is that when we go to look at where the ticks were found most frequently, it was within those more forested, more connected block types. And so we have kind of this connection here where the deer are selecting these forested residential blocks on Staten Island. And in fact, that's where the majority of ticks are being found in people's yards. So there's higher risk of exposure in those residential block types. And what we're working on now is simulating this deer space use so that we can have a visualization of tick-borne disease risk. So this would allow us to kind of develop expectations for tick distributions to be able to inform people within the community about where they should expect the majority of the risk to be based on the deer movement patterns that we've seen. So some takeaways here. We observe sex-based movement differences, and this can have implications for the distribution of ticks, where male deer may distribute feeding ticks outside of natural areas because they encompassed more of that urban landscape, while females stayed within the urban parks. So they may maintain the source tick populations within these green spaces. And then block characteristics that I identified with the residential areas did affect the probability of tick presence and survival in yards which interface with humans. So over 90% of the yards where ticks were present were in that forested block type that white-tailed deer prefer. And lastly, using these movement models to simulate deer movement in novel landscapes can be used to understand tick population dynamics, as well as more broadly to just identify areas where humans and deer are coming into more contact. So these human-deer interfaces. So this is not only important for tick-borne disease, but also important for diseases like SARS-CoV-2. Okay, so next I'd like to briefly introduce a second system that I'm beginning to work on through my postdoc. I'll only provide an overview because this work is in beginning kind of planning phases, and I don't have results at this point. So this work is examining how movement and contact between livestock and wildlife affect virus transmission in Nairobi, Kenya. So Nairobi is experiencing rapid growth of roughly 4% every year, and its population is estimated at about 8.5 million currently. 60% of the population occupies only 6% of the land, and that's residing in slum areas. And the slum population is expected to double in the next 15 years. Further, there's a large demand for animal source products, for dairy as well as for meat. And this results in really high urban livestock keeping in the city. So this makes Nairobi extremely different from New York City in that way. Livestock estimates from a publication in 2017 estimated over 700,000 livestock. So that includes cattle, goats, pigs, and chickens in the city. And these practices bring livestock into close contact with humans, making Nairobi an important area for zoonotic surveillance. So at a local scale, the growth in Nairobi is captured well by an urban gradient that encompasses both socioeconomic and large ecological variation. And this was established through previous work done by Dr. James Hassel at Smithsonian and the Urban Zoo team at the International Livestock Research Institute in Nairobi. The gradient here is basically describing how we see ecological change over alongside socioeconomic change. So at the top, as we move from the left to the right, we have high income areas that align with higher elevation, as well as high tree cover. In the high income areas, there's higher species biodiversity and lower human density and lower livestock density. And then in the low income areas on the right side of this gradient, there's higher human and livestock density, more artificial land use, so more impervious surfaces, and more synanthropic. The host community is more synanthropic dominated. So this is just species that live in close association with people. So here it's our rats and our raccoons. And so prior work sampled households across this gradient, and they found that the diversity of genes that encode for antimicrobial resistance, so this is AMR, in bacteria, that the diversity increased in seed-eating birds as household density increased, but only in areas and only in houses where livestock were kept. So this is that blue line going up here. And so wildlife that interact closely with humans and livestock waste within households may be exposed to more antimicrobial resistant bacteria. And this infers close contact between those seed-eating birds and livestock. So this close contact and potential for pathogen transmission is what I'll be focusing on with my upcoming work. And then further work from the same group examined multi-drug resistant E. coli across many different wildlife groups in Nairobi. So this map is showing an outline of the Nairobi extent, and the color is showing the proportion of wildlife with multi-drug resistant E. coli. So they found that fruit bats and seed-eating birds were significantly more likely to carry multi-drug resistant E. coli out of all wildlife that were sampled, and they sampled rodents and primates as well. So this is another motivator for my research, because these higher levels of multi-drug resistant E. coli can provide hints at the fact that wildlife and livestock may be coming into close contact. So here's one of the species that I'll be working with. This is the Epimorphus wahlbergi, the Wahlberg epilated fruit bat, and then house sparrows. And the main question that I'll be addressing with this work is, how does wildlife-livestock contact affect virus transmission in this rapidly developing city? Oh, and I would like to just show this video. This is from a site visit from last spring to a dump site to examine the marabou storks, because I am hoping to do some GPS tracking of their resource use across the city. So I'll be using these lightweight proximity sensors that are shown up here to collect high frequency contact data between the livestock species as well as between livestock and wildlife. And this will be including the Wahlberg epilated fruit bat, house sparrows, cattle, pigs, and goats. I'll also be looking at indirect contact through resources at households. So this will include things like manure piles or fruiting trees that may attract the wildlife and livestock to come into close contact. And then through collecting oral and rectal swabs, as well as blood samples from all the animals, these samples will be used for viral metagenomics to characterize the diversity of viruses that are circulating around the city, as well as look at the proportion of viruses being shared between these different species groups. And this is what's being shown here on the left, where we have kind of the social network where the wildlife, livestock, and resources are making up these nodes that are connected through contact. And then we'll have a viral community sharing network that will connect individuals with shared virus communities. So we hope to be able to identify these high-risk interfaces for pathogen transmission in Nairobi to provide information to the community and health officials and those kind of working in urban development on the ground. So please stay tuned for this work. There should be updates in the coming year and a half. Okay, so just to wrap up, urban zoonotic surveillance and prevention should be addressed with a multi-hazard lens. So we know that there aren't just tick-borne diseases circulating in New York, and if we're able to develop baselines for the distribution of these different zoonotic hazards that are coexisting, that can allow us to start to see changes in the distribution patterns of the pathogens and the hosts to be able to determine whether that change is of concern. So this figure is showing some coexisting data from New York City on the distribution and counts of rats, the same for ticks, and Aedes albopictus and Culex pipiens, 2 different mosquito species, alongside land cover and income. So how can we kind of integrate these different data sources to get a better understanding of how to manage these zoonotic hazards in cities? And then briefly, what can we do as individuals? So there's these different phases of pathogen spillover, and really I'm directing our attention here to this pathogen spillover phase, where we must continue working towards preventing deforestation, making urban growth more sustainable, reducing land use change from agricultural expansion so that pathogens that are circulating in nature remain contained in nature. And this isn't just for the benefit of human livelihoods, but it's also extremely critical for wildlife conservation and health. So, really kind of pushing this One Health lens as the solution in this way. Okay, thank you all for your attention. Oh, that was my dog. And I would like to thank all of the collaborators and funders who have made the work possible, but please feel free to contact me with questions or collaboration or funding ideas. I'll stop sharing now.
Thank you, Dr. VanAcker. That was somewhat sobering and fascinating. Hi, everybody. I'm Linnean Vice President Gabriel Willow, and pleased to join Dr. VanAcker for our Q&A. So I'm sure that was some food for thought. If anybody has any questions, please drop them into the Q&A box. You'll see a little button at the bottom of your screen that says Q&A, and you can type them in there, and I will relay them to Dr. VanAcker. We have one question already, which I'm sure is on a lot of people's minds, particularly if they live on Staten Island. or Westchester or something, which is it a bad thing to see deer or evidence of them in your yard or near where you live as it relates to ticks and whatnot?
Yeah, that's a great question because they're so ubiquitous in suburban areas. I think that it's important to kind of know that depending on the season, there may be more risk. So that might trigger you taking steps like assessing your landscaping, removing leaf litter that helps ticks survive over winter, and potentially seeking out companies that spray acaricides in the backyard, in your backyards, if you are seeing ticks. But I would say the first kind of logical move is to just take a piece of fabric and drag it along vegetation along the edge of your yard and see if you're finding ticks during their active periods. So that would be, start when the weather starts warming up. So anywhere from like April through August. And if you're not finding tick yourself, then there may not be reason to be concerned.
Now, when we have these fluctuations, which seem more and more the norm, someone who's a professor I know and a birder was noting in their Christmas bird count report that we've had this weather pattern for several years now where it gets a real cold snap in like early to mid-December and then a warming around New Year's, and it's often quite mild in January. At least that's been the pattern for a few years now in our region. When that happens, do the ticks kind of activate even in the winter sometimes if it stays above freezing?
Yes, yeah, they will be active on warm days, even in the winter.
Wow. So that's probably a piece of the puzzle as well. Another question we had that was my question as well, actually. People ask me a lot. So I lead bird walks and bird tours and stuff, and people often, sometimes you'll even find like unfortunately a deceased bird that's like collided with a window or something, and sometimes I'll collect them for New York City Audubon's Project Safe Flight. And people often wonder, like, is it safe to touch a bird? You don't know how it died. Could you catch something from it? And I usually tell people, look, wild birds don't spread disease to humans by and large. And, I'd wash my hands later. And that, like, I tend to think of most avian human diseases as coming from poultry, where they're in more close, contact and high concentration. But, how much zoonotic transfer or, spillover, I guess was the word you used, is there from birds to mammals? And is there less than, say, from rats or bats just because we're not as closely related?
Yeah, that's a big question because there's birds and bats are the two most important zoonotic hosts for in terms of the groups of species that we may see emerging pathogens from. And so I think it's kind of, it's a two-prong answer because you have the behavior of the animal, right? Like if you're handling a rat, you may be more apt to get bit. If you're handling a bird and you're trained in handling a bird, then that interaction may not be as risky, even though the animal may have some pathogen that could be transmissible to you. So I would do the same. I would recommend people wash their hands always after handling any sort of wild animal or carcass. And that the type of interaction that you're having with the animal also can determine the risk, even if they're a host, a reservoir host.
So there is some potential if one encounters a deceased bird for some disease.
I'm trying to think of an example of one that would still be active after death, but I'd have to get back to you. I'd have to get back to you on that.
Yeah, I tend to think not, but I don't actually know. I just wash my hands up. But yeah,
it's probably rare after they're dead.
Yeah. And now, of course, I keep hearing more about prions and like that, even picking up some, you find like a polished looking skull in the woods. And I thought, oh, you're safe there for sure, like a mammal skull. But apparently some prions can persist
For
in
years.
even for years. Yeah. So that's
in soil. Yeah.
nerve-wracking. Anyway, another question. Has your research shown any greater or lesser occurrences of pathogens in livestock as opposed to wild animals? Or have you, yeah.
I have yet to screen or sample the livestock yet. So I'm excited to do that, but I haven't gotten to answer that yet.
I guess from a fieldwork standpoint, working with wild animals is probably a little more exciting than hanging out with cows.
Yeah.
But more challenging too, perhaps. A cow, you can just walk up to it and take samples and stuff.
Yeah. More accessible.
Yeah. But less exciting. Now this makes sense. This is a good question. In New York City, given that these are the animals that people would encounter most often, is any significant disease being transmitted from rats, mice, raccoons, pigeons, or dogs and cats?
Wow. Yeah. So rats, there's leptospirosis where that's transmitted through rat urine. There are cases in New York City, usually a few every year. And so they're really low, but it's circulating and present. With raccoons, we have raccoon rabies, but that goes through, and actually, if you go to New York City Department of Health, they have publicly available positive rabies data over the years and what species they were found in. And so that has been found in raccoons as well as I think a few bats in the city. And the data is really accessible, which is really nice for the public. And we have canine distemper circulating through raccoons, but that's not infectious to humans. It's just going through the wildlife population. So there's definitely zoonotic pathogens that are circulating in the wildlife populations in New York. Yeah,
I think at some point there was a rabies outbreak among raccoons and they culled a lot of them, I believe. And then I think if I'm not mistaken, the Parks Department now or the Central Park Conservancy or somebody regularly will vaccinate the raccoons in the park against rabies, and they put a little metal tag on their ear so they know. Sometimes you'll see these raccoons with a little metal ear tag, and I'm pretty sure it's either for distemper or rabies, but I think rabies because that's more of a danger to humans.
Yeah, and they'll also do edible vaccines. I saw that a lot in the field, actually, when I'd be out sampling for ticks. You'd find edible vaccines in the forest floor that had been eaten. So they'll distribute the vaccines that way too.
Yeah, I just had my Flintstones rabies chewable. Delicious. Let's see here. What about, this is a good question. This is a fun one. Doing field work in New York City or, I guess some of it was in Staten Island, which is a bit more, I wouldn't say rural, but I know New Yorkers tend to see all kinds of things and not really ask too many questions and just go about their day. Not their problem. But yeah, did you ever get any questions or what were your human interactions like while you're doing field work within the city?
Really interesting. When I was when I showed the map of all the boroughs, I went to all the different public parks throughout all the boroughs, and so I surveyed Central Park. I got a lot of weird looks and lots of questions because I was wearing kind of the white suit and had the drag cloth. On Staten Island, we captured a lot of humans on camera traps, some that you didn't really want to see. And then, people on Staten Island were really inquisitive because it's, these parks are surrounded by their neighborhoods. And so, we'd give them kind of recommendations on how to treat their clothing and pulling their socks up over their pants, and really just where the risk was. If they lived in the north, they weren't, it wasn't as risky going into the parks as if you lived in the central part of the island. So it was nice having those sorts of interactions because we were able to kind of take what we were doing in the forest and relay it to the public. But I did, I met a guy who was out there searching for a diamond bracelet every day in the middle of a forest. So it was really interesting.
Had he lost it, or he just was hoping that he might find it?
Oh yeah, lost it like 12 years ago.
Whoa! Oh no! And he still was trying to find it. That's really sad. I hope he's probably out there. He's probably still out there. I hope he finds it. Somewhere there's a deer out there with a nice diamond bracelet.
Yeah,
that's fun. Here's a good question. Are flocking species like Canada geese, for example, more likely to serve as a reservoir or transmit disease compared to some loner species like an owl or something that's very territorial? And
that's a great question. I don't know if there's been work done at kind of a really broad scale comparing the type of behaviors in that way. But we tend to think that any sort of clustering enhances transmission. There has been really interesting work done on migratory species where they've found that infected individuals actually kind of fall off because they don't make the full migration. And so that can almost be a way of filtering out those infected individuals. But I don't know of a study that looks across kind of social dynamics like that. And that's a really good question. Yeah.
I guess that's an area for research for if there's an enterprising PhD student out there who needs a project.
Yeah, be a good review paper.
Sample a social species versus a non-social species and look for different diseases. I guess that could be pretty interesting. Now, someone requested for you to go over the tick life cycle again, like when are we most at risk? Does off or DEET or any of those things work well against ticks? And I know that opossums love to eat ticks. Are there any other critters to encourage that could reduce the tick population? So I guess that's like 3 questions, but they're all tick related.
Yeah, so the tick life cycle, basically the nymphal tick is the life stage where you're most likely to be bit. The tick has a high chance of being infected because it fed the year before and obtained infection. And if you're bit, it's still small. The tick is still small enough. It's the size of a sesame seed. So it's still small enough that you may not notice and it can fall off before you realize that you've been bit. And so the nymphal stage is the highest risk, and they're active in the Northeast from June through to August. So it's basically the summer. I would say actually their activity falls off before August, maybe like first week of August, end of July. And that's in terms of the black-legged tick. So you can be bit by the adult stage, but you're more likely to see it because they're much larger, and those are active in the fall. So the nymph is what you have to be most concerned with. And then DEET works. What we usually do is we'll treat our clothes with permethrin at the beginning of the field season, and that'll last for like 6 washes. So if you're active in the woods, pretty continuously through the summer, that might be a good option. And what was the last question? The last part of that?
Good question. It was, the animals that maybe can help keep tick populations in check, like opossums and things.
Yes. We captured opossums on Staten Island and they still had tick burdens. And so there was there's a few studies that showed that they may reduce the population because they eat them. But I don't think it's probably a strong enough effect that you would want to like introduce them into your backyard or anything like that.
Are there any other invertebrates or anything? I mean, I guess spiders might eat a tick. Is there anything much that could keep the tick population down?
People have also said, oh, those, I forget what they're called, the not quail. It's a similar.
Oh, guinea fowl. Guinea fowl.
Like yeah.
Yeah.
Yeah. People have also proposed that, but I don't know I don't know how those studies turned out and whether they had an effect.
And I wonder if they're eating. I mean, the nymphs are so tiny. I don't feel like they would be looking for those as a food source. It might be the adults. But I guess if they kill the adults, that's fewer eggs that get laid, and ultimately it may reduce the population.
Yeah.
All right. So everybody just has to get a bunch of guinea fowl and keep in their yard. They're very loud, so there's a trade-off there.
I mean, there's a lot of there's projects that are looking at vaccination for rodents because they are one of the primary reservoirs for the bacteria. That's going on in Westchester. And then 4 posters are another way of managing the tick populations from the deer side of things. So these are like feeding stations for deer. And when the deer put their head in, there's a caricide treatment that's put on their neck and around their ears and such that kills the ticks. And so there'll be, actually there's a project starting on Long Island and in different parts of the Northeast that will be looking at different intervention strategies like that. If
they could vaccinate rodents against it, there's no form of vaccine for humans against Lyme disease?
There's been development on it. There was one that was introduced historically, and then it was pulled from the market. There's also a vaccine for dogs. The difficulty with it is that there's different species of Borrelia. So Borrelia is very diverse. So it's hard to create a vaccine that's multivalent in that way, that it could capture all of the different types of Borrelia that are circulating.
So what we call Lyme disease isn't really one thing exactly.
It's Borrelia burgdorferi is the pathogen, but then there's different genospecies under that. And some are human invasive and some are not. It's kind of similar to how we treat SARS-CoV-2 in the way that there's different variants that are circulating, but we still call that SARS-CoV-2.
Right. Got it. Let's see. Oh, is there another? Oh, yeah. Now, a lot of Linnaean membership are bird watchers, of course, and birders always have a bit of a issue with feral cats. Are feral cats a disease vector or reservoir, and could that be another, angle to sort of try to encourage different municipalities to reduce feral cat populations, or like TNR programs and things like that sort of maintain these feral cat populations? Can they spread disease?
Yeah. So feral cats are a big problem that someone in the lab at Columbia works really closely on, actually. But they are definitely, they're reservoirs of disease. Wildlife disease. So the viruses that are transmitted between the cats themselves. They're also being screened for SARS-CoV-2 in the coming years. So that we'll have to kind of stay tuned to see what those results look like. But I think that it's definitely the disease component is an issue, and it's another reason to try and control the feral cat situation.
Good. All right. Next time I get in an argument with a feral cat person, I'll bring that up. That's right. I remember early in the pandemic, there's all these stories about. Just when you mentioned them being a reservoir for SARS-CoV-2, there was all these stories early in the pandemic of like this tiger at the Bronx Zoo that got COVID, or there was like, I don't know, a bear or something that had COVID. I haven't heard one of those stories in a while. I feel like we need more from the like zoo animals with COVID beat. That hasn't come up in a while. Maybe they all got vaccinated. I don't know.
Well, we're starting to try and look at SARS-CoV-2 in the white-tailed deer. So, and that may turn into more species in New York, but we'll see.
They're also a reservoir for COVID? Oh,
yeah,
boy.
yeah.
Wow.
Yeah, they've seen deer-to-deer transmission, and they've seen infection within deer throughout multiple different states now. So it brings another element to the deer movement focus, you know, because if we're able to identify some of these, like the reasons behind, the drivers behind how humans and deer interact, that is also important for potentially COVID.
Wow. You know, something I've heard is, you know, there's been this rise of everybody's favorite topic of bed bugs. And I've sometimes heard that link to the banning of DDT. Like DDT wasn't meant to control bed bugs; it was for mosquitoes. But because it was so prevalent in the environment, I had heard that, you know, maybe that's why there really weren't very many bed bugs around back in the 60s and 70s, and they've really the numbers have gone up in cities over the last 40 or 50 years. Do you think there's any link to that with ticks? I mean, because it does seem like they're increasing so much, and although there's like tantalizing suggestions of, you know, Lyme disease like diseases up, you know, 100 years ago or more, it seems like it wasn't really it didn't really pop up until right around the time that, you know, DDT had been banned. Do you think there's any link between the banning of DDT or some other pesticides and more widespread zoonotic diseases linked to invertebrates?
For Lyme disease, it's more linked to the reforestation of secondary forests and the deer population explosion. But Borrelia bacteria is ancient, and it's been found. Yeah, it was found in the archaeological dig that they did in Italy of the Iceman. So it's been circulating in nature for quite some time, but it really was kind of the perfect storm of reforestation of the forests, the increase of the deer population, and deer spreading into areas where they were previously hunted out. And then climate change is assisting all of these ticks in moving north. We found, you know, an invasive tick from Asia during the time that I was in my PhD on Staten Island, and that was likely brought over from some animal trade or animal transfer into New Jersey. So I think there's a lot of different things going on that are helping to facilitate the spread of ticks, but mainly it's you know, climate change allowing them to survive through the winter, move farther north, and the wildlife that are able to move them around. I mean, they're also moved by birds really long distances. They were moved into southern Canada that way, and so I think it's a combination of just ecological phenomena is kind of coming together at the right time, you know.
Yeah, I mean, this is totally anecdotal, but I grew up in rural Maine, and when I was a kid, never worried about ticks. I was out there, me and my dog, like out in the fields and the woods, and, you know, we would have known if there were ticks because we both would have had them, certainly my dog. Never saw a tick, and then now you can't walk out your front door without, I mean, there's wood ticks, there's black-legged ticks, there's even Lone Star ticks now, I think, and I assume that's mostly climate change because there certainly were deer and there was forest in that setting in rural Maine. So I think that up there, that it's all climate change, I guess. And they're saying there was a moose tick, this big tick that mostly feeds on moose, that just the numbers have been going up because more of them are surviving the winter, and it's really impacting the moose. Just because there's just the population would normally fluctuate and get knocked back every winter, but now they're just growing and growing in population. Yeah. Well, that's a bummer. I hope that Asian tick doesn't take hold. That's the last thing we need is new kind of tick.
It's a clonal species. It reproduces clonally. So the females, it's horrible. The females don't need males, so.
Oh no. I mean, good for them. That's awesome for them, but that's the last thing we need.
Yeah.
We had we have a question here. This might be someone you know because you share a last name. A past COO of the Detroit Zoo, or maybe it's a coincidence. Maybe it's just distant relatives. Past COO of the Detroit Zoo, and there's a vaccine for COVID in tigers that's being used in zoos, they say.
Oh, that's great.
So there we go. The tigers are safe. That's why we haven't heard the news stories about the tigers with COVID. I guess they are being vaccinated.
Yeah, that's great.
How about that? And then another question about the, referring to you mentioned earlier, migratory birds maybe being less of a vector, because if there's a sick bird, it might not complete its migration. And they're saying, would an infected migrant stopping prior to the completion of migration be the equivalent of like viral shedding? But I took it to mean that when you were saying it wouldn't complete the migration, it didn't just stop, but it probably would not survive, right?
It wouldn't survive.
And so at that point, it's not really much of a vector.
No, yeah. So the shedding would only be happening if they were alive and actively infected.
Yeah, that makes sense.
So, yeah,
they're weakened, right? They're already operating on such a sort of razor's edge of survival. If they're weakened, they're not going to complete it. Well, good. Then we can say at least migratory birds are not a major disease vector, hopefully. Yeah. Good. Well, that's a somewhat hopeful note. I think we'll wrap it up there, unless anybody has any pressing final questions. I'm not seeing any popping up. So I will say a big thank you to you, Dr. VanAcker, for sharing your research and your work. It's so important. Pretty cool, too, that you were already doing this research and work before COVID came along. And I'm sure there's a lot of interest and hopefully funding now, if there wasn't already. So keep up the good work. No
Thank
pressure,
you,
but it'd be great if you stop any future pandemics from happening. That'd be cool.
I'll try my best.
Yeah. So thanks, everybody. Next month, our speaker, our meeting will be on Valentine's Day. And our speaker will be, hang on, let me double check. I know he is from Fundación Hoko Toko. I'm just blanking on his name because I'm terrible with names. But it's going to be about Fundación Hoko Toko, which is based in Ecuador, and they do amazing conservation work there. And he is their conservation director. Still blanking on his name, but check the website and tune in. I won't be here. So I
David Parra Puente.
Thank you, Rochelle. Thank you. David Parra Puente will be speaking about the Fundación Hoko Toko on their the work they've been doing. So that should be great. Thanks again, Dr. VanAcker, and I will adjourn the meeting. And thank you to everybody who attended the meeting tonight.